Good morning, students. Today we are talking about xenobiotics. Competency number 7.5 is allotted for this topic. Here we are studying mechanism of biotransformation of xenobiotics and associated diseases. So these three terms are important: xenobiotics, biotransformation, and detoxification. First we will see these three words and then we will start the topic. First is xenobiotics. Xenobiotics are toxic compounds which may be accidentally ingested by us, or they may be taken as drugs, or some lipophilic compounds which are produced in the body during our metabolism. And next is compounds formed by bacterial metabolism. All these are toxic compounds and they are xenobiotics. The actual meaning of xenobiotic is strange substances. This xenobiotic is taken from Greek word. The meaning of xenos is strange. Now, what is biotransformation? Biotransformation is a process whereby a substance changes from one form to another form by chemical reactions occurring inside the body. In this, liver plays an important role. Now, during biotransformation, sometimes toxic substances are converted into non-toxic metabolites, or sometimes toxic substances are converted into more toxic metabolites. Depending upon this, biotransformation is of two types, and these two types of biotransformation are bioactivation or intoxication and detoxification. Bioactivation or intoxication, as the name suggests, in this case the chemical reaction occurs in the body which convert a toxic substance into more toxic substance, whereas detoxification means when a toxic substance is converted into less toxic substance. So biotransformation in our body may form more toxic substance, which is called as bioactivation or intoxication, or it may form less toxic substance, detoxification. Now these are the examples of bioactivation or intoxication. Here the vinyl chloride is given. Vinyl chloride is a toxic compound for our body because it is hydrophobic. Generally the toxic substances are the substances which are hydrophobic for our body, therefore they cannot be easily excreted from our body and they occupy a space inside the cell and they disturb the cell functions. So hydrophobic compounds are generally toxic for our body. Similarly, vinyl chloride is a toxic substance simply because it is a hydrophobic and which cannot be removed easily from the body. Now this vinyl chloride by biotransformation get converted into vinyl chloride epoxide in our body. Now this vinyl chloride epoxide, not only it is a hydrophobic, but it covalently bonds with DNA and RNA and causes mutation in DNA, which leads to cancer. Therefore vinyl chloride is converted to more toxic vinyl chloride epoxide. Similarly, benzopyrene, which is converted into a carcinogen by epoxidation of this benzopyrene. So these two examples are of bioactivation or intoxication. Now this is an example of detoxification. So if you see in this reaction, the toluene, which is hydrophobic, get converted into benzyl alcohol with the help of molecular oxygen and NADPH. Now this benzyl alcohol is partly hydrophilic because of presence of OH group, so it can be removed from the body through excretion in urine. Sometimes biotransformation may be dose dependent. The example of this is acetaminophen. This acetaminophen is nothing but it is an antipyretic drug. Now when this drug is given in normal doses, the body will convert this acetaminophen into harmless metabolite and it can be easily removed from the body when the action of this drug is over. Now if this drug is given in high doses, then the body will convert this acetaminophen into a harmful metabolite, more harmful metabolite, which is an example of intoxication. So the same compound or the same drug after its action may be converted into less harmful substance or may be converted into more harmful metabolites, and that depends upon the dose of the drug given to the patient. Therefore biotransformation depends on dose. Now which are the compounds which we have to detoxified daily from our body? Just now I have told you the compounds which are hydrophobic, they cannot be excreted through the urine or through the stools. Only hydrophilic or amphipathic, which have some part of or group attached to that compound is hydrophilic. Only these substances are removed from the body. Pure hydrophobic substances are not removed from the body. Now these are the compounds which are removed from the body, like compounds accidentally ingested, just like preservatives of food or food additives or food adulterants. Sometimes we take drugs in for therapeutic purpose. When these drugs perform their action, then they should be removed from the body. Generally the drugs are hydrophobic in nature, but after their action they should be converted into hydrophilic form and they should be excreted from our body. Now there are some compounds which are formed by bacterial metabolism. They are also harmful for our body, and the compounds produced during the metabolism of our body are also toxic for our body if they remain in the body. So these are the compounds we have to remove from our body. Now these are the examples of the compounds which are formed in our body during normal metabolism. First and most important example all of us know is bilirubin. Bilirubin is formed from breakdown of heme. When the bilirubin is formed, it is called as unconjugated bilirubin, which is not soluble in water. Therefore it cannot be excreted through the bile. So bilirubin is converted into bilirubin monoglucuronide and diglucuronide, and then it is excreted into the bile. Similarly, the amino acids which are present in our body, after their decarboxylation they form corresponding amines. For example, histidine is converted to histamine, lysine is converted to cadaverine, ornithine is converted into putrescine, tyrosine into tyramine, and tryptophan into tryptamine. So all these are called as amines, and these amines are also harmful for our body and they should be removed from the body. Now before starting the actual reactions of detoxification, you should know about this cytochrome P450 enzyme system. Now this cytochrome P450 enzyme system is responsible for majority of biotransformation reactions. Now why they are called as 450? Because they absorb light of wavelength 450 nanometer when they are exposed to carbon monoxide. Means these enzymes, the enzymes, it is not a single enzyme, it is an enzyme system. More than 150 cytochrome 450 enzymes are available in our body, and all they absorb light of 450 nanometer when they are exposed to carbon monoxide, and hence they are called as cytochrome P450. Where they are present, cytochrome P450 enzyme system is mainly present in liver. In the liver, they are found in endoplasmic reticulum. They are also found in adrenal glands. In the adrenal glands, they are mainly present in mitochondria and microsomes. Now, these cytochrome 450 enzymes contain heme and they contain phosphatidylcholine. Phosphatidylcholine means lecithin. It is a phospholipid. So, I have told you they are located in the endoplasmic reticulum of the liver mainly. Now, this is the mechanism of action of cytochrome P450. On the left side, I have written a hydrocarbon compound RH, which is purely hydrophobic in nature. Now, this cytochrome P450 will convert this RH into ROH, or it's converted into alcoholic form with the help of coenzyme NADPH and molecular oxygen. So here OH group is added. Due to addition of this OH group into the RH, it is now water soluble. Now this cytochrome P450 is also called as monooxygenase. Why it is called as monooxygenase? The monooxygenase are the group of enzymes which break the oxygen molecule and they will add one oxygen atom in one substrate. Here on the left side, two substrates are there. First substrate is RH and another substrate is two hydrogen atom donated by NADPH. So molecular oxygen is broken down into two atoms. One of the atom is added in RH to form ROH and another oxygen atom is added to the two hydrogen atoms obtained from NADPH. So molecular oxygen is broken down into two atoms and two atoms are separately added into two different substances. Such enzymes are called as monooxygenase. If suppose both oxygen molecule is added only in one substrate, then such enzymes are called as dioxygenase. Cytochrome P450 is a monooxygenase system. Here again the same example I have quoted here. Conversion of toluene, which is a purely hydrophobic compound, with the help of oxygen molecule and NADPH, it is converted into alcoholic form, that is benzyl alcohol. Almost all common drugs in our what we are taking are metabolized by cytochrome P450 enzyme system. Now this cytochrome P450, as I have told you, it is not a single enzyme. It has more than 150 isoforms. Isoforms means the proteins which are different due to their difference in post-translational modifications. So 150 isoforms of cytochrome 450 enzymes are found up till now. Now these cytochrome P450 are classified depending on their structural homology into family, subfamily, etc. For example, first you should know cytochrome P450 is abbreviated as CYP. Now I have given one example, CYP1A1. So CYP means it is cytochrome P450. First digit indicates family. Now second letter A indicates subfamily and the third letter again 1 indicates the first member of that subfamily. So in this way, just like enzymes, they are named. Now next example is CYP4D3. Here CYP indicates cytochrome P450. 4 means it belongs to family 4 and D means it belongs to family D in that family, and 3 denotes that it is the third member in that subfamily. So in this way they are named, no necessary to remember here. Just you should know whenever short form like this is there in your textbook. Now the most important property of cytochrome P450 enzyme system is they are inducible enzymes. Inducible enzymes means the enzymes of which the concentration can be increased or decreased. So such enzymes are called as inducible enzymes. On the contrary, the enzymes for which the concentration inside the cell remains constant throughout the life, they are called as constitutive enzymes. Now, cytochrome P450 are inducible enzymes, and the drug phenobarbital causes increased activity of all cytochrome P450 enzymes. What will happen? So, when a patient taking phenobarbital, this phenobarbital induces the cytochrome P450 enzyme system. Therefore, the activity of cytochrome P450 enzyme system increases, and as I have told you, this cytochrome P450 involves in the biotransformation of various drugs. So, in such person who is already taking phenobarbital and their cytochrome P450 is induced, if we take a drug, the biotransformation of that drug occurs in that person at a faster rate. So this is the graph, shows you the biotransformation in two persons. On the y-axis I have taken antibiotic level in the blood, and on the x-axis I have plotted duration in hours. Now two graphs are there, graph 1 and graph 2. So if you compare this graph 1 and graph 2, it is found that in graph 1 and graph 2, in both person we are giving the same amount of antibiotic dose. But in graph 1, the antibiotic level is coming to 0 earlier as compared to graph 2. So this graph 1 is a patient who is taking phenobarbital, and graph 2 is a patient who is not taking phenobarbital. That means if a patient is taking phenobarbital, his blood level becomes lowers earlier as compared to normal person. Therefore, you have to adjust the dose of a person who is taking phenobarbital, all drugs. So this phenobarbital not only affect a single drug, all other drugs, antibiotics, antipyretic, antihistaminics, whatever drugs are there, all drugs are detoxified faster as compared to normal. As they are detoxified and removed from the body faster rate, you have to give more doses, or you have to increase the frequency of doses in such person. Sometimes due to phenobarbital, untoward effects may occur without taking any drug. For example, phenobarbital or barbiturate induces a enzyme ALA synthase, delta aminoleuconic acid synthase. Now this enzyme belongs to heme synthesis pathway. Now when this enzyme is stimulated, more and more intermediates of heme biosynthesis are synthesized and they get accumulated in the body, and this leads to a disease called as acute intermittent porphyria that all of us know. It is a disorder of heme synthesis pathway. So this is also sometimes a bad effect of the drug phenobarbital that you have to keep in mind. Now we will study some drug reactions of phenobarbital or interactions between the phenobarbital and other drugs. So first and most important example is warfarin. Warfarin is a anticoagulant drug. Generally it is given to a person after major surgeries like cardiac surgeries like angioplasty, open heart surgery, or when foreign body is kept in our body. To prevent the coagulation, we are giving this warfarin drug to the patient, but at the same time the patient is taking phenobarbital for other disease. So what will happen? This phenobarbital will stimulate the enzyme CYP2C9. It is a cytochrome P450 enzyme, and this enzyme is responsible for metabolism of warfarin. If a patient is taking both warfarin and phenobarbital simultaneously, due to stimulation of this CYP2C9 or induction of this enzyme, the warfarin is metabolized at faster rate than normal, and such persons will have to give more warfarin doses, because the warfarin doses given in the normal doses are not sufficient for this patient, simply because they are detoxified at faster rate. Next, about ethanol. Now ethanol is a alcohol which induces CYP231 cytochrome P450 enzyme, because it is involved in the metabolism of that ethanol. Now this cytochrome P450 not only metabolize ethanol, but it is also responsible for activation of some carcinogens in the body, and hence it is said that the patient taking ethanol have more chances of carcinogenicity, especially the liver cancers, because of this activation of this cytochrome P450 enzyme. In some persons, some of the isoforms of enzyme cytochrome P450 exhibits low activity, which is called as polymorphism. Means some of the enzymes out of 150, some of the enzymes of cytochrome P450 have less activity. So what will happen? Such persons were detoxify the drugs at slower rate, and this explains the variation in drug response among the different persons. That means for this person the normal doses are also higher, because the drug is metabolized, or blood will remain in the blood for a long period of time, as they are metabolized at slower rate due to decreased activity of some of the enzyme of cytochrome P450, and hence the doses of the patient you have to adjust according to the person. Now this graph shows the person, normal person, and the difference between the normal person and the person with low activity of cytochrome P450. Again, on the y-axis I have given antibiotic level in the blood, and in the x-axis duration in hours. Now we have given same dose of drug for both normal person and the person with low activity. Here graph 1 is of normal person, and graph 2 of the patient with low activity of cytochrome P450. If you compare these two graphs, then we can find out that in graph 1 the antibiotic level decreases faster than graph 2, or you can say in graph 2 the antibiotic level in the blood remains for a long period of time. In such person we have to reduce the doses or frequency of doses to avoid the side effect of the drug. That point also you have to remember in our mind. Now one most interesting example of cytochrome P450 is CYP2A6 enzyme. Now this enzyme is responsible for conversion of nicotine into a metabolite, and that metabolite is responsible for addiction towards nicotine. Now this enzyme CYP2A6 is synthesized by 3 alleles in our body. In some persons all these 3 alleles are inactive, therefore this enzyme is not synthesized in the body. So what will happen? The nicotine is not converted into its metabolite, and such persons do not show addiction for nicotine. If in some person 1 or 2 alleles are active, then they will synthesize less enzyme, and it will convert less nicotine into its metabolite, and this person shows less addiction towards nicotine. But if all 3 alleles are active, then they will convert nicotine into metabolite in large proportion, and this metabolite shows, and due to this metabolite formation, the person have strong addiction for nicotine. Therefore, you can see in our society some patient have addiction for nicotine and strong addiction, some have less addiction, and some have no addiction towards nicotine. Now cytochrome P450 enzyme system present in adrenal gland is responsible for steroid biosynthesis. It is not for it is not for biotransformation. Now we will start actual process of detoxification. Detoxification reactions are generally classified as phase 1 and phase 2 reactions. Phase 1 reaction is alteration of the foreign molecule by adding some functional group. The first phase 1 reaction may result in detoxification or intoxication. In phase 1 reactions, the functional groups are added to the foreign molecule by the process of oxidation, reduction or hydrolysis. Now after addition, these substances or the products of phase 1 reaction may either excreted or they may enter into the phase 2 reactions. The product of phase 2 may become the substrate for phase 2 reactions. Phase 2 reaction generally involves conjugation with conjugating agent. Sometimes the products of phase 2 may undergo phase 3 reaction, but it is a rare. Now we will start phase 1 reaction, and the first reaction of phase 1 is oxidative reactions. They are also called as hydroxylation or dehydrogenation. Now these are the examples of the compounds which undergoes oxidation in our body. Compounds with methyl group get oxidized to alcohol. Aromatic and aliphatic alcohols are converted into acids. Aromatic hydrocarbons are converted into phenols. Aldehydes are converted into acids. Anilides are converted into phenols, and amines are converted into acids. So these substances undergoes oxidation. Now coming to the actual example, again the same example I have quoted here. Toluene is converted into benzyl alcohol with the help of oxygen molecule and NADPH. So here the compound with methyl group is converted into alcohol. So methyl group of toluene is converted to CH2OH, that is alcoholic group. Second example is conversion of benzene to phenol. So aromatic hydrocarbons converted into phenols or alcohols. The oxidation and detoxification of alcohol is also important in the liver. All of us know. First, the alcohol is converted into aldehyde form with the help of enzyme alcohol dehydrogenase and NAD as a coenzyme. Later on, this aldehyde is converted into acid with the help of again enzyme aldehyde dehydrogenase and NAD. So this is the example where ethynyl alcohol is converted to acetaldehyde with the help of enzyme alcohol dehydrogenase, and then acetaldehyde is converted to acetic acid with the help of enzyme acetaldehyde dehydrogenase. Both these reaction requires NAD as a coenzyme. Now next, in the lower side I have given benzaldehyde, which is also converted to benzoic acid. So these are the examples of reactions in which they undergo oxidation, they undergo detoxification by oxidation. Sometimes oxidation may result in intoxication. For example, when methanol is undergoing oxidation, it forms formic acid, which is more harmful than methanol. Similarly, ethylene glycol is converted into oxalic acid after oxidation. So these are the examples of intoxication after oxidation. Now the next reaction is reduction. Reduction is generally done by addition of hydrogen atom or hydrogen atoms. The compounds which undergo reductions are aromatic nitro compounds, they get converted into amines. Some aldehydes and some ketones get converted into alcohols after addition of hydrogen atoms. Now these are the examples of reduction. Here nitrobenzene is converted into amine, that is nitro compounds are converted into amines with the help of NADPH. Similarly, the picric acid is converted to picramic acid and paranitrophenol is converted to para-aminophenol. So when you are writing in the exam, no necessary to draw the structures, just only write down the names which compound is converted into which metabolite. Next reaction is conversion of chlorol to trichloroethanol. Again you have to write down here NADPH is converted to NADP, which is the hydrogen donor. After completing the reduction, now coming to the hydrolysis. All of us know hydrolysis is a chemical reaction in which addition of water split the toxicant into two fragments or smaller molecules. Here toxicant is a xenobiotic. It is denoted here as AB. Now the hydroxyl group is added to one of the compound and hydrogen atom is added to another compound. Here what will happen, basically the water is broken down into OH and H. Then the xenobiotic or toxic compound is also broken down into two small fragments. For example, A is converted to A and B. Now OH group is added to one of the smaller fragment and H is added to another fragment. So in this way hydrolysis takes place. Large amount of compounds are hydrolyzed by this process. For example, esters, amides, hydrazines, amides, glycosidic bond, peptide bond, ester bonds and carbamates. Now this is the first example of hydrolysis where acetinolide is converted into aminobenzene and acetic acid, and I have told you the substrate is broken down into two by addition of water molecule. Next example of hydrolysis is conversion of acetyl salicylic acid, which is also called as aspirin, into salicylic acid and acetic acid. The third example is conversion of diisopropyl fluorophosphate into hydrogen fluoride and dialkyl phosphate. So these are the examples of detoxification by hydrolysis. Now something about aspirin. It is a very widely used drug in clinical practice. It is analgesic, antipyretic and anti-atherogenic. Analgesic means it is a painkiller, antipyretic means it decreases the fever, and anti-atherogenic means it decreases the thrombotic property of platelets, or it will not allow clot formation. So this is aspirin. Now phase 2 reactions. In phase 2 reaction, generally the conjugation takes place. The xenobiotic, majority of the xenobiotic of phase 2 reactions come from the product of phase 1. The xenobiotic that undergone phase 1 reaction is now a new metabolite that contain a reactive group like hydroxyl group or amino group or carboxyl group. These metabolites undergo additional detoxification reaction in phase 2 reaction by conjugation. Conjugation also make the compound non-toxic into easily excretable form. Now before starting conjugation, you should know the conjugating agents. These are the conjugating agents. Glucuronic acid, the active form is UDP glucuronic acid. Active form is the in which form it participates in the reaction. Next is sulfate. Sulfate can also act as a conjugating agent. The sulfate which participates in the reaction is called as PAPS, phosphoadenosine phosphosulfate. Cysteine. Cysteine is donated by glutathione. Glutathione is a tripeptide which is made up of cysteine, glutamate and glycine, 3 amino acids. Now some amino acids like glycine and glutamine also act as a conjugating agent. Methyl group act as a conjugating agent, and the active form of methyl which participates in the reaction is called as S-adenosyl methionine. Acetic acid also act as a conjugating agent. The active form of acetic acid is acetyl-CoA. Now we will study one by one examples of these conjugating agents. Now first is glucuronic acid as a conjugating agent. The glucuronic acid, active form of glucuronic acid is called as UDP glucuronic acid, which act as a donor of glucuronic acid. Now this UDP glucuronic acid combines with a xenobiotic from phase 1, that is ROH, and transfer is glucuronic acid to form RO glucuronide. The enzyme responsible for this transfer of glucuronic acid is UDP glucuronyl transferase. It is present in the endoplasmic reticulum. Glucuronic acid also conjugate hydroxyl groups, carbonyl groups, sulfhydryl groups, and amino compounds. For example, phenol is converted to phenyl glucuronide, benzoic acid is converted to glucuronic acid monobenzoate, bilirubin, all of us know bilirubin is converted to monoglucuronide, means one glucuronic acid is added, or some two glucuronic acids are added to the bilirubin, then it is called as diglucuronides. The steroids are, after conjugation with glucuronic acid, forms glucuronide of steroids, amines forms N glucuronides. So these are the compounds undergoes detoxification by conjugation with glucuronic acid. Next example is sulfate as a conjugating agent. Now sulfate will conjugate with phenolic compounds and alcoholic compounds. The donor of sulfur in the reaction is active sulfate, which is called as phosphoadenosyl phosphosulfate, and the enzyme sulfotransferase will transfer the sulfur from active sulfur PAPs to the xenobiotic. Now here ROH is a xenobiotic which have again come from phase 1 reaction, react with PAPs, and sulfate group SO3 is transferred to this xenobiotic so that it becomes more soluble in water. ROH is less soluble, whereas ROSO3 is more soluble in water, so that it can be removed easily from the body. Similarly, phenol combines with active sulfur to form phenyl sulfate, and indol combines with active sulfur to form indoxyl sulfate. Now important compounds excreted as their sulfates include steroids and indol compounds, so they are excreted into the urine. Next, cysteine as a conjugating agent. Now here free cysteine is not available in the body for reaction, therefore glutathione is a active cysteine, or it can donate cysteine during the reaction. Now by conjugation with cysteine, the following compounds are detoxified. First is alkyl halides, aryl halides, epoxides, and alkenes. Now this is the mechanism of action of conjugation with cysteine and detoxification. Here the compound is RX, alkyl halide. Now this alkyl halide combines with GSH. GSH is called as reduced glutathione. Now this enzyme glutathione transferase will transfer the hydrogen atom from glutathione to the halide X, which forms XH. Now after removal of hydrogen atom, the reduced glutathione GSH is converted to GS, and that oxidized glutathione is transferred to the group R. Now in the second step, the enzyme GGT, gamma glutamyl transferase. Now this enzyme will remove the glutamic acid from the oxidized glutathione and transfer it to an suitable acceptor. Therefore, in glutathione only two amino acids are remaining. They are cysteine and glycine, to which R group is attached. Next, enzyme peptidase act in between cysteine and glycine. Cysteine and glycine both are amino acids, so they contain peptide bond. So enzyme peptidase will break the cysteine and glycine bond, peptide bond, and transfer the R to cysteine to form R cysteine, and glycine becomes free. Now this R cysteine then sometimes combines with acetyl CoA to form mercapturic acid by enzyme N-acetyl transferase. So in this way, the cysteine can act as a conjugating agent. Now conjugation with glycine. Here the example is benzoyl CoA combines with glycine to form hippuric acid and coenzyme A. Now this benzyl CoA generally we are taking as a food preservative. They are added in the food as a food preservative, which is a hydrophobic compound, therefore it is difficult to remove. In the liver, this benzyl CoA is conjugated with glycine and converted into hippuric acid, which is water soluble and easily excreted out of body. Next is conjugation with glutamine. Now example of this is phenylacetic acid, which combines with glutamine amino acid, and complete glutamine is added to it to form phenylacetylglutamine. Therefore it becomes more soluble and easily excreted. Now next example of detoxification by conjugation is through methyl group. Methyl group can use for detoxification of amino group, hydroxyl group or thiol group. And as I have told you, the methyl does not participate on your own. So yes, adenosyl methionine, also called as active methionine, because it is participating in the reaction, act as a donor of methyl group, and O-methyl transferase is the enzyme which transfer the methyl group from SAM to the xenobiotic. Now these are the examples. Here first example is phenylepinephrine. Phenylepinephrine combines with S-adenosyl methionine. Methyl group is transferred to phenylepinephrine to form metaepinephrine. After donation of methyl group, S-adenosyl methionine is converted to S-adenosyl homocysteine. The enzyme is catechol O-methyl transferase. Next example is conversion of pyridine to N-methyl pyridine. Again, SAM donates methyl group in the presence of enzyme catechol O-methyl transferase, and SAM is converted into SAH. Next example is mercaptoethanol, in which the SAM donate methyl group to form 5-methyl mercaptoethanol. The enzyme is same, catechol O-methyl transferase. Sometimes this methylation causes intoxication. Instead of detoxification, it causes intoxication simply because methyl group is a hydrophobic group. When it combines with mercury, it forms methylated mercury, and this methylated mercury is more toxic compound as it causes damage to our nervous system. Next example of detoxification by conjugation is through acetylation. Here the donor of acetyl group is acetyl CoA. The enzyme responsible for acetylation is N-acetyl transferase. The compounds which undergoes acetylation reactions are generally drugs like sulfanilamide, isoniazide and para-amino-salicylic acids. So this is an example of sulfanilamide drug. Here acetyl CoA donate its acetyl group in the presence of enzyme N-acetyl transferase, so that sulfanilamide is converted into acetyl sulfanilamide. So it can be easily removed from the body. Next example is conversion of isoniazid to acylated isoniazid. Again acetyl CoA donate its acetyl group in the presence of enzymes N-acetyl transferase. So this is about the various conjugation reactions or phase 2 reactions. After phase 2 reactions, I have told you some of the very few metabolites enter into the phase 3 reaction. Again, in the phase 3 reaction also conjugation takes place, and here the conjugation takes place with glutathione. No example is quoted for this in your book, so no necessary to study the enzyme of this class 3 reaction or phase 3 reaction. After completing the biotransformation, you should know what is the significance of biotransformation. The first and most significance of biotransformation is biotransformation convert most of the toxic substances into non-toxic substances, that is detoxification. But at the same time, second significance is you should remember sometimes due to biotransformation, the toxic substances it converted into more toxic substances. That you have to remember in your mind. Now third significance is during biotransformation reactions, if the cytochrome P450 enzyme system is induced it may create untoward effect. For example, induction of ALA synthase by barbiturate will lead to synthesis of more and more intermediates of heme synthesis, which get accumulated in the body, leading to acute intermittent porphyrias in some patients. Now, biotransformation is also important, particularly to study the effect of drug, which varies from person to person. As I have told you, some person shows lower activity of cytochrome P450 enzyme system due to genetic variation. This may lead to decrease or absence of expression of enzyme activity, and some people who can metabolize the drug sluggishly may show toxic manifestations with normal doses of drug. In such person, we have to adjust the doses according to the patient's need. So this is about the biotransformation, xenobiotics and their disease. Thank you very much.